Okay, I guess now we are. Okay, and how many people stuck with me? Oh God, 29 out of 30 stuck with us. Okay, did you think of any questions in the meantime? I want to be able to handle all the questions right now before we try to link over to the share screen. If we get the share screen, we're never going to turn it off today. Okay, and I'm going to call the IT and find out uh, why it's doing this. I tried. I was using Firefox. I thought, oh, that could be a problem. So I switched to Chrome, as you're supposed to be using Chrome. Didn't work. Went back to Firefox, and finally got it to work. But everything was so slow. Okay, here we are. Got any questions for me?
Is there a separate Zoom meeting for the bio discussion class? Like,
Yes.
okay.
Hang on. Uh, Considering what just happened today, okay, the bio 110 discussion is 1 hour a week. Okay, you would think that the smart thing to do maybe is a half hour today and a half hour on Thursday, but the state of California does not allow half-hour classes. That's why it was put on one day, and the other day was left blank. Well, I told you what I'm doing with the blanks. We'll just start and keep going till we finish and forget the blanks. You know, take a break when we need to. I want to do the. I'm set for the 110, ready to go, but I'm going to do it on Thursday. And how am I going to do it on Thursday? We're going to have our regular, hopefully, question and answer, but then our lecture, then our lab, okay, and then I will do it afterwards, okay, and then if you now the 110 is meant to help you. It's meant to give you even more review, and I've got some really good review pictures set up for you. It's also there to ask more questions, although today we're probably questioned out. So I want to be sure we get through it without problems. So lots of times these problems originate from the school itself, and then they go away quickly, like in an hour or so. But I don't want to mess with that today. So I'm going to do the 110. I'm going to do 1 hour on Thursday after we're finished with everything else, not today. Okay, so we get our time in. I have to do it in the 110 area because they do monitor how long you stay in each area, and they don't care. They just do things like that. You know, so yeah, it will be there. It is ready for you. Okay. Now, if you're not enrolled in 110, and 6 people are not, you still can if you wish. And you need an ad card. All you have to do is email me, and I'll send you the ad code, and you get it done. You get till February 6th, I believe it is. If you don't ever want to join the 110, for whatever reason, the $46 that it costs, or you just don't think you need extra review or whatever, fine. I will. I still have to go ahead with it for the people that signed up, and I want to. Okay, but you won't be able to get into it. You won't have an access code. Uh, so, how do you get the information? Well, maybe you didn't want it in the first place, like I said. Otherwise, you could ask somebody to record it for you while they're in there. Okay. I'm going to put it on YouTube, and if you want to try to, go ahead and find it on YouTube or ask me. I can do it the roundabout way, I suppose. But I'm not trying to upset the people who enrolled. They, okay. So Thursday's your answer. Is that okay, Haley?
Yeah, you said Thursday, right?
Thursday. And normally we finish our work at 4 o'clock, 4:15 sometimes. So on Thursday, this one time for the class, you would meet and probably stay till about 5. Now, as far as attendance is concerned on the 110, okay, I'm not going to be counting attendance. I'm going to be counting the chapter outlines that are due and seeing that they're all done on time. Okay, that's the way for an A in 110. The way to get benefit of further review is to stay for the 110. Okay, I'm not going to time you in, not going to time you out or anything like that. Okay, I don't want 110 to be a hardship. I want it to be a help. That's the intention. I will also have a practice test for you to take, see how you're doing. Now, one thing about practice tests, I noticed last week, and I wasn't going to say it until we got there, but we haven't gotten there yet. I want to say it now. The practice test at the end of week 1 was taken by 11 people. Only about a third of the class took it. That's the most important thing you can find to click on so that you can find out how well you're doing, how far behind you've already become in 1 week, okay, and what you need to do to catch up and stay up, okay. It's recorded by how many people took the test, which questions they may have missed, how many times it took them to get that high score. The average score was 92, but that was only after several tries, okay? But needing several tries means you learned. You learn from taking several tries, okay? It also does report it by your name, okay? Which test you took, how many times you took it, each and every question you answered. Too much information, okay? That practice test is for you. If you don't take the practice test itself, well, good luck. You could have done better, I'm sure, on the exam. Okay, I really do want to see the practice test be taken by the other 20 students. Okay. It will help. So, Andrew Brenner, are you?
Yep,
I don't.
we're here.
There you are. Okay. Yeah, you were asking me, is the practice test like the regular lecture test? Definitely it is. Okay. And who wouldn't want to see it? I can't understand why 20 people didn't bother to look at it. Okay, we're recording, you know that. What happens if I share the screen since you don't have any more questions, I take it? And we get lost again. If we get lost again, go ahead and sign off. I'll get back on and I'll send you an email. I will send you also the pages that we would have covered today. I don't want to do it that way, okay? But I don't want you to feel that you're getting behind because of technology here. This is the way I've always entered share screen. So let's give it another shot. Okay, there we are. Can you see this here?
No.
No?
No.
Okay, well, I'm looking at choices. And the choices is the screen. And that's the one I always click on. Down here, the other corner, there's the word share. But that only works after you've gotten into it. So there it goes. And
We see
now I try to log into, can you see yourself over here?
You can see your screen.
Yeah.
You can see the screen.
Yes.
We see the launch meeting screen.
Yeah.
Yeah.
Yeah,
Okay.
we can see it.
I don't know that we can go from here. If we can, that'd be great to find a shortcut. Log in again. Okay. Return to meeting. This is the way I've been having to do it. I go to Saddleback College and I log in. There, let me do it this time. No rhyme or reason. I really want to thank you for hanging in there. Okay, I'll go faster here, but that was great. Okay, this was last week. At the bottom, Charlie's website again. I'll probably move that right now down to here, so it keeps following us. The recording from last Thursday. And then here's the YouTube of the skull. This is, if you didn't look at it yet, this is really very good. I don't like listening to lectures myself, but this guy was pretty good. Okay, here we are this week. You have a chapter assignment. The chapter assignment is chapter 9. You only get one assignment this week. I gave you some examples already. I gave you a handwritten example, which is fine. These chapter assignments, these 3, 1, 6, and 9, are not due until P1, Practical Exam 1. And that's on February 10th still. That's when they're due, February 10th, the P1. Okay, the shortest lecture topic there is, levels of organization. You read about this already. Okay, it's labeled lecture so Joe is to distinguish it from the lab material. The lowest level of organization is the cell itself. And you're going to say, wait a minute, what about a molecule or an atom or an electron? Yeah, well, those are subatomic or whatever. They're not living. Okay. Cell, generally considered the smallest living entity, or the smallest living thing. The truism is that all cells come from pre-existing cells, and all organisms are composed of one or more cells. To say all cells originate from one or from a pre-existing cell cannot be true when the first cell was formed, because where would the first cell come from? Not another cell, because that would be the first cell. So the first cell didn't. But today, we're talking about 2000. All cells come from pre-existing cells. And all organisms are composed of cells. If we say one cell, that would be like a paramecium or an amoeba. If you're talking about yourself, you're in the more cell category. A tissue, the definition of which would be asked of you several times, is composed of similar cells that perform a common function or function. Okay. A common doesn't mean commonplace. Common means the same function. For instance, we have 4 basic tissue types. We have a lot more tissues. We have a lot of very finely named tissues. But when we put them into categories, there are only 4 main categories of tissue. The first is the epithelial tissue. Epithelial tissue is tissue that covers or it lines. Okay. Covers something. Well, the skin covers. The outer layer of the skin is stratified squamous epithelium. Don't hurry to put the word down. You'll get plenty of time for that. Okay. Or it lines something, the lining of the stomach, the lining of the blood vessels. Okay. the skin covers the body. And on one side is the body, and the other side is the air, the environment. Okay. We have nervous tissue. I don't think we need to say anything about that now. Muscle tissue. And then connective tissue is the fourth category. Connective tissue connects or interconnects structures or connects them chemically. Chemically meaning by, for instance, blood. Bone is a connective tissue, so that's what we've been studying so far. Blood, adipose, cartilage, and there are other connective tissues that are going to be given to you. Many others when we get to histology. Right now, just understand that it connects or interconnects. Pictures of the 4 different types of tissues. These are really more artist conceptions. When we have tissues that work together to perform a common function or functions, they're called organs. And when I ask a class in face to face and I put it up on the board, name me some organs. Oh, this is the one where they can come alive. Everybody knows organs. So common is to see the stomach, the pancreas, the heart, skin, liver, brain. Can you name me one more organ?
Kidneys?
Exactly, kidneys. Is it one more? Somebody else? What about
Lungs?
what?
Lungs.
Lungs, that's the one I was just going to say. And the gallbladder, okay, and then the large intestine's not here. The small intestine's not even here. This is just a short list. Of this list of 6, which 2 organs do you think use up the greatest amount of calories, of energy in a day?
Stomach.
Brain.
Okay, brain is correct. What's
Heart.
the other? What? Heart?
Heart.
No.
Stomach.
No.
Lungs?
Lungs.
The lungs
Liver.
aren't here.
Liver.
Liver. Yeah, you finally whittled it down to there's only one left, right? That's great. The bottom 2, the liver and the brain, use up most of the energy. They go constantly at the same pace, day and night. Of course, if you skip down and read this next sentence, it would have told you that. Organ systems are when you have groups of organs that do the same thing or work together to do the same thing. And this was the 12 black and white photographs that you saw. And I told you not to worry about at that time. But now, nervous system, muscular, integumentary, the skin, digestive, respiratory, reproductive, lymphatic, circulatory, endocrine, and urinary systems are the vein systems of the body. Okay, you can say systems, or you can say the whole thing and call them organ systems. So as you see, we are getting more and more complex as we go up. And when all the organ systems are put together, we have the organism itself. Not difficult. Okay. We need to do something now that most of us, you and me, would consider remedial. There was never a biology class that didn't put a picture of the cell on the board and have you label it. I just can't believe that. And we need to go over the cell. Why? Because we have forgotten a lot of the terms. It's amazing that on the first lecture exam, most of the questions missed are on the cell. And I tell people it's going to be there. You probably have forgotten some of it. Please review it. And I think they think that they must know it. My feeling is we get to where we are based upon what we once knew and may now have forgotten. I mean, do you remember your civics class in 10th grade? Do you remember anything about? I don't remember a damn thing. Okay. Had to take it though in order to get to the next grade. Okay. So I'm going to go through this pretty darn fast because we're a little behind. If it's too fast, you just tell me. Cell structure. Big word, cytoplasm. Cytoplasm is all the material inside of the cell membrane. Here's the cell membrane here, also known as the plasma membrane or plasmalemma. The cytoplasm is all the material, fluid and solid, between the plasma membrane and the nuclear membrane. That'll include such organisms as the mitochondria. We'll define it a little further on. The peroxisomes, the rough endoplasmic reticulum, we'll define. Ribosomes, vacuole. Centriole is the preferred word here. Microtubules, lysosome, Golgi apparatus, Golgi vesicle, which becomes the lysosome, smooth endoplasmic reticulum, the nuclear area itself, chromatin inside of the nuclear membrane, but not this dark spot, which is the nucleolus. Does anybody, I'm not going to call on you, I'm not going to embarrass you or anything like that, but does anybody feel that they know most of these terms?
Yeah, kind of.
Good. Okay. Anybody else? I'm trying to get a feeling for how fast I can go through them.
Yeah.
I know you can read it anyway. Okay. Inside of the nucleus, there is a dark spot, okay, that we can see with a light microscope. That's the nucleolus. The nucleolus. Its definition is down here. It's a condensed area of material within the interphase nucleus. An interphase nucleus is what we're picturing here. An interphase nucleus is not dividing at this point. It's not dividing in half. A lot of people used to say the interphase nucleus was asleep. Nonsense. Or dormant. Nonsense. This is the most active time for a cell to be an interphase. Okay. It's getting ready to divide. The nucleolus is believed to be responsible for the formation of transfer RNA. We are not going into protein synthesis and how it's accomplished. That's not a part of gross anatomy. But I will simply tell you that tRNA, ribosomal RNA, and messenger RNA, okay, are the three types of RNA that we recognize, and tRNA is produced. We believe in the nucleolus. That's surrounded by the nucleus itself, or nuclear material, and especially the chromatin. Chromatin is only seen in the interphase nucleus. Where are the chromosomes? They are spread out. They are spread out. If you take a piece of plastic rope, you've seen plastic rope, I believe. It comes yellow and blue, usually down at the shipyard or something. And you twist it against itself, you can open up the plastic. You can open up the fibers far apart, and they get really less dense. That's the chromatin. That plastic rope, when tightened, represents a chromosome. The genes on the chromosome are not exposed when the chromosome is tightened. When it's loosened, then the genes are exposed and you can get the formation of messenger RNA. This area around dark, what color is that?
That's purple.
Purple. These little purple dots are really tunnels interconnected with each other. And the dots that are on them are ribosomal RNA. As material leaves the nucleus and enters the rough endoplasmic reticulum, which this is called, the rough endoplasmic reticulum manufactures enzymes, proteins. All enzymes are proteins. And that continues and seems to fan out. We have it from left to right, but it's towards you and away from you as well, in all directions. These enzymes, and then they reach an area where no more enzymes are formed, called the smooth endoplasmic reticulum. Here, the enzymes accumulate, but are not being formed further. They accumulate, and they can form packets. These packets can join and end up forming this structure down here called the Golgi apparatus. Used to be called the Golgi body, still is. And then coming off, almost like a space station here, sending off messenger vehicles. Our small Golgi vesicles, we don't need that term because they enlarge quickly by taking up water and form lysosomes. The enzymes that are formed from the rough endoplasmic reticulum are proteolytic enzymes. They break down other proteins. They're packaged up in the Golgi apparatus and sent off as lysosomes to look for material to digest and to release those nutrients into the cell, to look for bacteria, to look for anything that the cell has brought in, such as a broken red blood cell, for instance. Okay. Sometimes these are called suicide packets because some believe that as people age, the lysosome attacks the cell that it's in, loses its direction, if I can say it that way. Okay, let's look at some of these terms. The cytoplasm, it's the area outside of the nuclear membrane. It's all of this, liquid and solid. The nucleoplasm is the area inside. Chromatin is the DNA or chromosome that is unraveled during interphase. By being unraveled, it's able to be used to form messenger RNA for the production of other structures. The nucleolus is here, the dark spot. This will disappear during division. Some organisms have more than one, we just have one. Nucleolus produces T or transfer RNA. The nuclear membrane around the nucleus, but it's continuous with the rough endoplasmic reticulum. This is all two-dimensional, of course, but picture yourself with a bowl or a colander of cooked spaghetti. You're draining it. Those strands of spaghetti don't just go right and left. They go up and down all over the place. And so do this. So do these. You would think inside of a cell, things can float pretty darn freely. You know, how? You're going to be bumping against endoplasmic reticulum everywhere you go, which is pretty much going to actually hold you in place. The centriole is located here. This will split in two during division, and its function when we get to division will be repeated. It's to set up the spindle apparatus to help direct the motion or movement and splitting of chromosomes. The endoplasmic reticulum is divided into two parts, the rough with the ribosomes and the smooth endoplasmic reticulum. Protein synthesis takes place in RER. I always ask this question. And protein packaging is more limited to the SER, smooth endoplasmic reticulum. The Golgi apparatus would be next. There's no vacuole shown yet. The lysosome I showed you here contains proteolytic enzymes again to break down whatever the cell brings in. The mitochondrion, I always ask this question. Mitochondria, what's their function? They are the center of cellular respiration. Not breathing, respiration, but you break down material to ATP and NADPH, energy useful materials. Okay, we didn't do vacuole, but we're going to now, along with phagocytosis and pinocytosis. These two things, phagocytosis and pinocytosis, can be abbreviated if you want to talk about them both at once as cytosis. This is cytosis. We'll learn soon that this is an active process of the cell. You've all looked at a golf ball, whether it's miniature golf or professional PGA stuff, I don't know. But you've all looked at a golf ball and you see the indentations all the way around. How many of those little indentations are on one golf ball? I had a guy in the class once who knew this stuff. It's ridiculous. What clouds your mind with the fact that there are 237 on most golf balls of those indentations. Well, on the golf ball, the indentations, the roughened surface, is static. But on the cell surface, those indentations pull in fluid and minerals, that's the star. Okay. It's also called solids here. Or they can pull in minerals, fluids, and bacteria or broken red blood cells. The one on the left, the situation on the left where the cell membrane is constantly doing this, sucking in, brings in mainly fluid. The solids dissolved in the fluid, we still call that fluid. But when there's a larger particle brought in, it's a little bit different. The process is called pinocytosis when fluids are taken in. It's called phago, meaning to eat, when larger particles are brought in. Pino means cell drinking. Pinocytosis is cell drinking. Phagocytosis is cell eating. Whether you're bringing in fluids or you're bringing in solids, the lysosomes will be looking for these to break them down, release the nutrients into the cytoplasm, and let the mitochondria respire and form energy-giving compounds. Now, in the pinocytosis situation, the vesicle that is formed is called a pinocytic vesicle. Here's the term up here. And when in phagocytosis, we have what is called simply a food vacuole. Okay, it's 3:19. Let me give you to 3:30. And then I'm going to go over the next set of bones there, and you'll find that they're much friendlier than the skull. Okay, so stop and see you at 3:30.
Thank you.
Okay, back. I have an email address for everybody now. Some did not send me their preferred one, so they got their school address. If something comes to you and that's what it comes as, and you don't want it, still tell me. Okay, but I got some of the personal addresses when people wrote me a couple weeks or so before school started to save everything. Any questions?
I actually have a question, but it's kind of more general about the anatomy courses at Saddleback. I was just curious why we don't use the cadavers at our school and we use cats instead.
We don't use many cadavers. We have 4 cadavers, and we bring them into the room. You identify muscles on them. The reason we don't use more is years ago we got our first one that we had to dissect ourselves. It took them a year and a half to do it because students did it, but students kept crapping out. I'm sorry, don't mean students are crappy. I mean they kept flaking away. They didn't want to finish it, and so we got to find new recruits and more, and it just takes forever to get one of them done. So at most schools, I believe, not just Saddleback, but at most schools, they will have them prosected, professional, you know, and that's fine. But for what use we get out of them, looking at the muscle tissue, we went past that. We start tearing it apart, and it's useful just one time.
Mhm.
So we do use it for the muscle, the second session.
Okay.
I don't know that there would be much of a use after that. I was in charge of a class once at UCI, and UCI had only 1 cadaver, and that happened to be a woman. Very unusual that she was only 30 years old. Usually the cadavers are very old, having died of a stroke or congestive heart failure. Uh, if they die of something else, it usually has to have an autopsy to prove it wasn't poison or this or that. And once they're autopsied, they're of no value to anybody. So that was our situation. I led a class. They tried to dissect, but you can't watch 30 people at one time. And while one person is doing it right, the so next group is just slicing away and making a mess of things. So we have it done professionally. And remember at UCI thinking, oh God, I wonder how many people are going to get sick when they see the cadaver. And I was in an inside hallway, bam, the door from the laboratory would open up. It did. And it was a guy come out and he just unleashed all over the floor. It was sickening. It was really sickening. So was a guy. So was a guy too. But there was one girl or woman. You know, I'm not allowed to say girl, but I'm going to do it anyway. I can only say female and woman. I'm not cannot say ma'am. I can't say gal, of course, or anything like that. What can they do to me? I don't know. But this one young woman, I'm not even allowed to say that. she was so interested in the cadaver, and she really did great work. The cadaver had a pillowcase on its head because we did the facial muscles last. When you unmask the cadaver, it becomes more personal, and nobody was having any trouble except that one guy, but he was in a different class. She was asking me, this one girl that was really good, said, "What are all those spots on her arms?" And I said, "They're freckles. They're just like yours." She was redheaded. And she said, "Oh no, they don't look like my freckles." I said, "Freckles are different. You know that. You know that they're different types." Anyway, we took the mask off on the last day to do the face muscles, and not only did she have freckles, this could have been that girl's twin sister, and she just had a hysterical fit and ran out of the door. Never know what you're going to get. Okay. Any other questions? I don't mind these general questions at all. I feel we get to know each other a little bit, and know a little bit about the program. Okay, let's go on. Okay, here we are. We're back. And this time, I'll point out this thing here, bones and figure locations in the text. This is just a guide again. You open it up, and it tells you the name of the structure. This is your list, this is your study guide, but it tells you what figure in the book you'll find this, so that you can look at it when you're on your own. Okay. Yes, we have pictures that are labeled as well, but if you want a specific one, here's how to find it. Sometimes I think it's valuable for you to bring the bone figures and locations with you. In other words, print them out, have them while we're looking at the bones themselves. And when do we look at the bones? Right now. Bones and structure, the vertebral column and the ribs, labeled photos, and it's part of the lab. Okay. Okay, this is a typical vertebra. A typical, well, we have different types. We have cervical in the neck. We have thoracic in the chest region. We have lumbar in the small of the back. We have the sacrum, and then we have the coccygeal vertebra, or the so-called tailbone. This one is a thoracic vertebra. Okay. Viewed from the side or in profile here, the thoracic vertebra has a fairly big head. I call these ears, but then the tail really slopes down. You can't see that slope when you're looking from the top here, but from the side you can. This is the dead giveaway that you have a thoracic vertebra. There are 12 thoracic vertebrae. Okay, we label them T1 through T12. Simple as that. The front or forward part is the body. Here's the body over here when we're looking at it from the top. Let's look at it from the top now. Okay, body held up by two pedicles. A pedicle. Two structures come out. These are the transverse processes. It doesn't say that here. It says it over here. Transverse process. A part of a rib will attach to every thoracic vertebra. That's the other distinction. The head of the rib will fit in here, and it swings back and touches the end of the transverse process at what is called the transverse facet, or just facet. A facet is a face. And then the rib swings around and goes forward around the central part of the body. Back to here. Pedicle, transverse process. Next part is the lamina. And here's the lamina over here. Sometimes when people get pinched nerves, as they're called, and end up with back problems and such, an older technique that's still used is a lammectomy, and they cut this. If they botch it and cut what's in here, which is the spinal cord, they can do more damage than good. Lamina, lamina, and here going down is the spinous process. This is going down and away from you because it's this over here, the spinous process. Sticking up, straight up here and here, are these two structures, here and here. These are the superior, because they're on top, articular, because they are joints, processes. This is the superior articular process. This is the other one. Here the two are. And they each have a facet or a face. What they make a joint with is the inferior articular process of the vertebra that is above them. Here's the inferior articular process over here. Now it can't fit up here at the same time, so it fits on top of the one below it. And they make a fairly nice chain. You can take your little finger, or the tip of it anyway, and put it into this hole. That's the vertebral foramen. Actually, most people can't fit their little fingertip in there, just the tip, can't fit their little finger in there. Okay, going around one more time, we'll start at the spinous process, to the lamina, to the transverse process with its facet, transverse facet. We'll skip this for the time being and go to the pedicle, and then the large structure, the body, and all of this surrounds the vertebral foramen. The structure we skipped was this one, the superior articular process with its facet. If I were to ask you this, the superior articular process, I would definitely either point properly or point to the facet. Or I might word the question, name this process or name this facet, so that you are not confused as to which one I want. At the top, here's the first cervical vertebra. It's called C1. C2 is underneath it. Do you remember the occipital condyles? The two bumps on the underside of the skull. Those occipital condyles fit here and here. This is C1. You can almost stick your thumb through this one. Why? Because it doesn't have a body. It doesn't have a body. There's a reason for that. Here's the transverse process. Transverse process looking different. Here's the spinous process. I would never ask you to describe the lamina versus the pedicle. I think they're a little bit too obscure. Actually, this one looks pretty good. Only in the cervical vertebra do we have these holes. This is a transverse foramen. The vertebral artery goes down through that, and nerves go down through that, through your neck, and they're semi-protected by going through there. We don't have this hole in the transverse process of the thoracic or any other vertebra. The second cervical vertebra is the axis, whereas the atlas allows you to nod your head yes, up and down. The axis fits in here, or put it another way, the atlas C1 fits on top of C2. And this structure called the dens comes up here and allows you to twist your head right and left in a no gesture. Here's the transverse process. Here's the transverse foramen. Here's the spinous process. There are 7 cervical vertebrae in your neck. C1 is the atlas. You need to associate that name with it. C2 is the axis. You need to associate that name. C3, 4, 5, and 6, the only thing special about them is to see that they got the transverse foramen, because only cervical vertebrae have it. These 2 are distinct. No body and the dens. The others are just pretty typical. But that's through number 6. Number 7 is the bump at the back of your neck, down at the bottom of your neck. You feel that bump, okay? I'm gonna trust that most of you are actually feeling that bump. That's C7. It's known as the vertebral prominence. I'll see where I've got it in the handout here. Vertebral prominence, the prominent vertebra. But before we go there, this is a lumbar vertebra now. Did some cervical, did some thoracic, and now here's the lumbar vertebra. These are the largest vertebra. There are 5 of them. They're L1 through L5. And the muscles of the legs are lower body muscles attached to their transverse process. Here's the body. Here's the transverse process. Here's the superior articular process. Here's the inferior articular process. And back here, the spinous process. Rather than looking like a spine as it did in the thoracic vertebra, the spinous process in the lumbar looks like you had a piece of clay and you squished it between your thumb and index finger. Flattened it. Here's the sacrum from the posterior side. This is not anatomical position. Anatomical position would be to look at it from the other side. The base of the sacrum is actually this entire structure at the top. You see a triangle in front of you, obviously, and that portion is the base. When we get to the heart, the base will be similar to this. There's a crest in the middle, a raised portion, like jagged little mountains. This is the mid-sacral crest. It has other names: a middle sacral crest, median sacral crest, sacral crest. I like mid-sacral crest, and that's why I put that one there. come down. Here's the exit for a lot of the spinal nerves. And this is the sacral hiatus. It's continuous if you were to take a applicator stick or a little stick and push it up underneath this to come out up here. This is the sacral canal, sort of the equivalent of the vertebral canal. But it goes through not just one bone, but 5. Comes out at this point, the sacral hiatus, and at this point we call this the apex. The base with the star, the apex of the star. The holes almost always number 8. Sometimes it's not the case, sometimes 10, sometimes only 6, but almost always 8. These are the posterior sacral foramen. The spinal cord actually finishes off at L2. We can't see it here. But fibers come down, nerves come down, and exit through these 8 openings. Posterior sacral foramina. The tailbone, as we call it, is the coccyx, coccygeal. Originally, the tailbone might have consisted of 4 pieces, but they fuse together as you mature. The sacrum may have consisted of 5 pieces, but it's now 1 as you mature. Here's the anatomical view. You're looking at it from the front now, as if you could look through everything that would be in the way. At the very top of the sacrum is the sacral promontory. This is important in childbirth because if it sticks into the pelvic region too far, the kid's head's not getting through. And if the kid's head's not getting through, you're not going to have a live birth unless you have a C-section. There may be other ways of doing it that I don't know about. I'm not an obstetrician, obviously. These 4 holes on each side, total 8, like on the other side. These are the anterior sacral foramina. It looks like there are 8 holes on this side and 8 holes, and they go through to the other side. But these 8 here are not the same as these 8 here. They are holes in the plate, in the bone, and they match up with each other. So the total number of holes is really 16. 8 on this side, 8 on the other. Here's our sacrum again. On the edge here, it has a place to articulate. This is the auricular surface. Here it is, turned to the right. This sacrum, turn it to the right. I'm going to look at it from this end. It's this. This is the auricular surface. Auricular means ear shaped. Somebody's got a good imagination. They're calling that an ear shape. So that's the auricular surface. It's attached to a wing of bone right here. It's just the side of the wing of this bone. This bone is called ala, A-L-A, meaning wing. Okay. Here's the ala up here, when I didn't mention it, and here. The auricular surface is on the side. finishing off the axial skeleton. The axial skeleton, I told you the other day, but I'm sure you didn't remember it from that, has 80 bones, and these bones are part of that. First, the sternum. This is the sternum. It's sectioned into the upper part, the manubrium. Looks like a shield. Then the body, and then the xiphoid process. This little piece that hangs down. You're all familiar with the xiphoid process. You pushed against it all your life when you're bored in lecture or whatever. Okay. When somebody does CPR, they don't want to be pushing down on the xiphoid process because they can break it free. That makes it very difficult for you to maneuver. You'll survive it, but the idea is you don't want it. Okay. Here are the ribs. A pair of ribs, number one, here's the one, attached by way of costal cartilage. What color is this, please? Costal cartilage.
Like a blue.
Okay, go with that. This is the costal cartilage. And a pair of ribs attached to the manubrium. Then you have attachments for rib 2, rib 3, the pair of rib 4s, 5, 6, and 7. Okay. 8, 9, and 10 do not have their own attachment. They piggyback on number 7. And 11 and 12 ribs don't have any attachment at all. The first 7 pairs, this is where a sheet of paper might come in handy. You might want to consider it in the future. I can always repeat anything, and you can also get it, of course, off of the recording. 7 pairs of ribs, it's 14, or what we call true ribs. These are the true ribs right here. The false ribs are the last 5. Because they don't have their own connection. And the 2 that have no connection, ribs 11 and 12, aren't, they are also false ribs, but they are also called floating ribs. Some people have had the floating ribs removed to make their waistline thinner. It was a rumor for years that Cher had it done, but she did not. Other people, though I'm assured, have had it done. It sort of ticks me off that a person skilled enough to remove those ribs isn't fixing cleft palates or something instead. Okay, you shared another difference between a true rib, false rib, and the floaters, or floating ribs. Here's the end of a rib. You don't get to see the extension of it. This first part is the head. It attaches to a thoracic vertebra on its side. Then a skinny part, the neck, and then a bump called the tubercle that articulates with the transverse facet of the thoracic vertebra. Look, 1, head, neck, tubercle. Head, neck, the neck is in here, I'm sorry, tubercle. And then it swirls around on both sides. Okay. Head, neck, tubercle. Then we have the body or the shaft. So when you actually feel your ribs or put your hands against the sides of your chest, you're feeling ribs, true ribs for the most part. And this is the body of your shaft. On the underside, there's a indentation, an indentation. This is the groove. It's not all that important, except if you want to identify the rib as a left or a right. This is a left rib. Head, neck, tubercle, swing out to the left because the groove is at the bottom always. If the groove were at the top, we have the rib upside down. We'd have to turn it. Here's our manubrium again. But now at the top, we have the jugular notch. You can feel your own jugular notch. If you were going to have a tracheotomy, that's where it's going to go. On either side of the jugular notches are the two clavicular notches, here and here. And that's where a portion of the clavicle, which we haven't done yet, okay, is going to fit in. Clavicle fits here, clavicle fits here, goes that way, and goes that way. Most commonly broken bone in the body. You would think it's the humerus. And if I asked you if you have broken bones, you would come up with an answer that would indicate the clavicle is not the most broken. It is though, when you take into consideration older people. They fall more often than you do, put their hands out in front of them, and when they do that, that's just ripe to break the clavicle. Here's the manubrium. I won't ask you sternal angle. Here's the body. And here are little facets, little holes, where the costal cartilage of the true ribs fit in. If you've ever had a tooth out, even your first set of teeth, there's always a hole left, at least temporarily. And these are facets for the attachment of costal cartilages. It's not coastal, it's costal. Okay. Anybody with any final questions?
I don't have a question, but like I have a way to remember like how many like vertebrae are in each group, like lumbar and whatnot.
Okay.
So like for the clavicle, we eat breakfast at 7, then we eat lunch at 12, dinner at 5, and we have 2 desserts for the last part. And then that's how I learned.
Okay. I don't know if I'll remember that because it's 7 o'clock. I'm not even awake. I go
Yeah.
to bed. I go to bed about 4 o'clock in the morning. I didn't used to. I used to have an 8 o'clock class and I had to be awake. But ever since this has started, this pandemic, I've been just going to bed later and later and later. Well, I'm sure we've all changed somewhat. Okay, thank you, Julia.
Hopefully
Anybody
we'll
else?
someone.
Yeah. Yeah. Okay. Okay. When I send you your next email, that you know that'll be pretty soon. Probably after I've got the video ready. Then just check the email address that I'm sending it to, and you're reading it off of. If that's what you want, great, because that's the way it's going to stay. Otherwise, okay. And we need to get going. I'm very happy that almost everyone stayed with us today throughout all that. Okay, so that we don't get behind. That's great too. Okay. Please do that practice quiz if you have it. Okay, you'll be grateful to me. Well, maybe you won't, but I think you will. Okay. Okay then. Go ahead and exit if you wish. Stay if you want to ask me anything.
Thank you.
Bye-bye.
Bye. Bye.
Bye-bye.
Professor, I had a quick question. So as far as the, what was that other one, the Bio 110 class, is that going to be, is that just during this class period, or is that like going to be a separate thing?
No, normally this class, I'm able to do it 1 hour lecture, okay, and the first hour lab, like a lab lecture, and then you would be on your own. I'm going to continue on after another break, okay, and then it'll all be continuous. It'll be at the end of Bio 11 first, Bio 11 lecture, Bio 11 lab, and then 110. 1, 2, 3. Okay. You should still be out by 5 o'clock if we don't run into any problems like we did today.
Gotcha. So it's kind of in between the lecture and lab is what you're saying?
No, nope, I'm not going to do that.
Oh, okay.
If I did it between the lecture and lab, the 6 people, you know, if they didn't want to sign up, that's fine, or couldn't sign up or whatever. But I'm not going to put the 110 in between, because they're going to sit around and do what for an hour? Nothing.
Right.
I don't want to waste people's time, and it'll be the same amount of time for you. So it's lecture first, lab, and then 110. Okay. And I invite people to 110. You don't have to go to it. You paid for it, but you don't have to go to it. If you have questions, especially go to it. And like I said, I have additional models, images, labels, stuff to help you out with. Okay.
Okay, thank you.
I missed that. When do we start the second class again? When does it start?
Oh, we're going to do it on Thursday.
On Thursday? Okay, cool.
Yeah. Yeah, but it's 1 hour a week, period. That's all it's supposed to be. And I'm going to do it on Thursday this week. If I feel like it, or maybe because we have a test coming up and it's more, makes more sense to do it on a Tuesday to prepare for a test, I'll tell you that. We'll do it on a Tuesday. We can play this one by ear. I don't definitely want to schedule it except at the start of the week. Okay, or even on the weekend, I can involve you and tell you this week we're going to do 110 on Tuesday, so that you can get prepared.
Okay, one more question. Are we going to be going into like what the cell looks like, an interface and all that stuff?
Yes, unfortunately. I mean, I don't like to do it myself.
Yeah.
Over and over again. But me, I've done it so many times. But a lot of the people in the class did it once, and then have forgotten it. And I don't blame them. You often forget.
Yeah, I forgot most of it.
Yeah, you don't even need it, so you tend to forget it. But you are going to need it for when we do histology. So that's why we do it. And that's why I finished that cell off pretty darn quick today. Anybody?
Is that exam 1? Is it going to be an exam 1?
Yes.
Oh, okay.
Everything we do until lecture test 1 will be on lecture test 1. Okay. Sometimes I do not include the lecture immediately before a test so that people don't have to cram, and it'll be on the next test, but you'll know well in advance of that.
Cool.
Sorry, Professor, I had one more question.
Sure.
It's actually about the anatomy quiz 1. Is that okay to ask that here? Should I wait for the review session?
You can ask it here.
All right.
It might be. Are you going to be in the review session?
Yeah, I'll be in there.
Okay. Why don't you ask it there?
Okay, cool.
I'd like it to, spread out to more people.
Okay, sounds good.
Okay. I mean,
Thank you.
if you're dying for it, you can ask me, but you can email me too. But I'd really appreciate, any questions you have that affect everybody. We ask them where it's best.
Okay, thank you.
Thank you.
Thank you.
Thank you, Charlie.
Okay. Take it easy, Joshua.
Hi, Charlie.
Hi.
Did the class end? Sorry, my laptop
Yes.
died before, and
Well,
I didn't charge it.
some people are asking questions, but yes,
Oh.
it's over.
Oh,
We're
okay. I'll look over the video once it's
Okay.
uploaded then. Thank you.
Bye-bye.
Bye-bye.
Ellen, are you still here? Caleb, are you still here? Jasmine.
Still here.
Okay. I was just asking before I cut you off. I didn't want to do that. Okay, so you got a question or anything?
I do. I just on the skull, I'm looking at it in the notes, because there's the temporal bone, and I know that it says squamous portion and petrous portion. And I am having trouble differentiating that between the mastoid process, if that makes sense. Like, it looks kind of this, like, is it the same thing, or am I just misunderstanding it?
They're all on the same bone. All the things you mentioned, they're all on the temporal bone. The point behind your ear, can you feel it? The bump behind your ear, that's the mastoid process. It hangs down from the temporal bone. Okay. The temporal bone itself is up here. Can you see where I'm touching? And that's a flat portion of it. So that's called the squamous portion. But
Mhm.
below that and down here, the mastoid process is on a not flat part of the bone. And that stylomastoid process is not. And let me get you a picture. I didn't look at all of this guy's video. It was pretty darn good though. So I'm gonna see if I can cruise to it. Oh, I don't want to see that movie. That's the trouble you get with. Oh,
So is it correct if I say that the mastoid process is on the petrous portion of the temporal bone?
absolutely.
Okay,
Okay.
so, okay, I think what happened is, I get it then, because I watched the video and I did look at the notes, but for some weird reason, because I printed the PowerPoints, I have a lot of visual trouble with things, so I was looking at the mastoid process from a different angle and I just wasn't sure, but that makes sense. So it's all on the same portion.
You see this
Mhm.
view down into the skull? Okay, see this structure here?
Mm, I don't see anything.
Uh-oh, did I cut us off? Not
Oh no,
sure.
I see you. I just don't see like whatever you're looking at. But
Oh, I'm looking at from this point all the way back to here, down here, same thing. Well, if that's not working, I'm not concerned actually, because I'm going to show it to you many more times.
yeah, no, but that makes sense. Thank you for clarifying because I thought it was on the same portion. I just wanted to make sure that when I look at it from different angles, I'm understanding where everything is.
Okay,
Thank you.
okay, I don't know if I've cut us off or not, but you got your question answered, right? So, goodbye.
Bye. Thank you. See you Thursday.